Okay, so welcome back to this next video in which we are discussing protein folding and protein folding diseases. Okay, so we're currently in the process of discussing gain-of-function protein folding diseases, which remember are when the protein misfolds and then gains some new function, generally that it aggregates now, and it's the protein aggregates that then cause the disease phenotype. Okay, so we've now looked at the AL amyloidosis or primary systemic amyloidosis, and we've also seen these three examples of transthyretin amyloidoses. Okay, what we're now going to move on to is discussing another amyloidosis, okay, and this is dialysis-related amyloidosis. Okay, so dialysis-related amyloidosis is going to involve yet another protein, okay, and this protein is going to be the beta-2 microglobulin of the MHC class 1 molecule. Okay, so what I'm going to start off with then is a little bit of a reminder of the structure of the MHC class 1 molecule so that we understand what beta-2 microglobulin is. Then we'll say what happens in renal failure and how this beta-2 microglobulin protein builds up in renal failure, and then we'll discuss how dialysis can then cause this beta-2 microglobulin to then start forming amyloid fibrils, particularly in the bone, which leads to bone erosion. Okay, right, so dialysis-related amyloidosis then. So we need to firstly look at the MHC class 1 protein complex. Okay, so this is an extremely important protein complex in immunology. It stands for the major, that's the M, histocompatibility, that's the H, okay, and then the C is for complex, so histocompatibility, and then complex, and this is the major histocompatibility complex class 1, so there is also the major histocompatibility complex class 2. Okay, now, the major histocompatibility complex class 1 is made up of two separate polypeptides, okay, that are joined together in a non-covalent fashion. Okay, and this complex is expressed on the surface of pretty much all cells in the body. Okay, so let me show this now. So, if we have our cell membrane here, and I'll just represent the cell membrane as one, actually I'll do it as two lines now that I've said that. Okay, so there's our cell membrane as a phospholipid bilayer. Okay, and there are two polypeptides that are going to make the MHC class 1 molecule. Okay, the first is what's known as an alpha chain. Okay, and the alpha chain has three important domains here. Okay, so here's the alpha 1 domain, and here's its amino terminus. Then following the alpha 1 domain, what you're going to have is the alpha 2 domain here, still extracellularly. Then you have the alpha 3 domain here. Okay, and then you have a portion that crosses the membrane, and then the C-terminus is on the intracellular side. Okay, so this is what's known as the alpha chain of the MHC class 1 molecule. So let's highlight this up. So here are our three alpha chain domains, alpha 1, alpha 2, and then our alpha 3 domain there. So this is our alpha chain. Okay, then, attached to the alpha chain, in this remaining little space here, you have the beta-2 microglobulin. Now, this is not attached covalently to the alpha chain, okay, so its interactions with the alpha chain are non-covalent. Okay, so here is beta-2M, which stands for the beta-2 microglobulin, and this is the other separate protein which makes up an MHC class 1 molecule. Okay, so I suppose I should probably just give you a little taster of what the function of the MHC class 1 protein is. So as I say, this is expressed on the surface of pretty much all nucleated cells in the body. Okay, and the function of it is to put little fragments of proteins on the surface of cells. Okay, so all the time what is happening is if I draw a little cell here, the cell is taking cytoplasmic proteins from, okay, so proteins from the cytoplasm, it's processing them, okay, it's cutting them up into little fragments, and it's putting these fragments in this binding pocket between the alpha 1 and alpha 2 domains. So there is a little binding pocket here where you can put protein fragments. Okay, and it's putting these protein fragments on the surface. So it's continually taking samples randomly of proteins in the cytoplasm, cutting them up into little fragments and expressing them on its cell surface. Now, what's the point of this? Well, normally, if the cell is perfectly healthy, all it will be doing is displaying little fragments of normal, healthy proteins. Okay, and this will not trigger an immune response. Okay, no cells of the immune system will start attacking the cell when all it's got displayed on its surface is MHC class I molecules with normal protein fragments in. Okay, however, if the cell gets infected with some intracellular pathogen, for instance a virus, notably viruses are the main ones which MHC class I is targeting, okay, what will happen is if I draw the virus now inside the cell, so let's say here is a virion particle that's now infected the cell, okay, what will happen is that virus particle here will drive the cell to start producing viral proteins. So the cell will start producing loads of viral proteins, and these are abnormal proteins. So let's say these red proteins here are the viral proteins. Okay, but the cell will continue on doing this process. It will continue on sampling proteins randomly, and eventually it will end up sampling some of these viral proteins, putting little fragments of those on its surface in MHC class I, and that will then be recognized by the immune system. It will say, these are not normal fragments. This cell is infected, let's destroy it. Okay, and that cell will be killed by the immune system. Okay, to protect all the other cells. And you might say it's a little harsh on the cell, but remember, there's 100 trillion cells in the body. Sacrificing one cell to protect all the others from getting infected with the virus is worth it, as far as the multicellular organism is concerned. Okay, right, so that's the function of MHC class I. It's very, very important in the immune system. It's present on all cells in the body. Okay, right, now, what can happen is the beta-2 microglobulin can dissociate away from the alpha chain from time to time, okay? So MHC class I molecules, they're not perfectly stable. Eventually, the beta-2 microglobulin might just break away from the alpha chain and go off into the extracellular fluid, okay? And it's not attached to the membrane, remember, it's only attached to the membrane because it's attached to the alpha chain. So once it's dissociated away from the alpha chain, it will go off. And then eventually it ends up in the blood. Okay, so this is normal physiology, by the way. Okay, this isn't pathology yet. So beta-2 microglobulin will end up in the blood. So let's have our bloodstream represented here. And now what removes it from the blood? You can't just let it stay there and beta-2 microglobulin will gradually build up and up and up throughout your life, okay, until your blood is just made up of beta-2 microglobulin. No, it has to be removed. So what removes it? Well, it is usually removed and broken down by, well, it's either the liver or the kidneys, and it's the kidneys in this case. Okay, so the kidneys remove the beta-2 microglobulin and break it down. Okay, so it goes to the kidneys for catabolism. Okay, now, this presents a problem if you have got renal failure. Okay, so if you've got renal failure, then your kidneys are not functioning properly. They're not removing waste products out of the blood in the way that they should, okay? So lots of your kidney cells are dysfunctional or dead, basically. And now the kidneys are not functioning properly, and that's known as renal failure. Okay, now, what's that going to cause for the beta-2 microglobulin? Well, it means that beta-2 microglobulin is going to build up in the blood. And you might say, aha, that is why we're going to get amyloidosis now. Okay, the beta-2 microglobulin in this really high concentration is now going to misfold and it's going to form amyloid fibrils. Well, actually, no. It's more complicated than that, and this is why it's called dialysis-related amyloidosis. It really is related to dialysis. Okay, beta-2 microglobulin turns out to be a fantastic protein as far as not forming amyloid fibrils is concerned. You can take this protein and put it in massive concentrations in test tubes, and it does not form amyloid fibrils. It is a very well-designed protein that does not form amyloid fibrils, even in enormous concentrations. So the enormous concentrations that you find in people's blood with renal failure, those are not what cause this protein to form amyloid fibrils. The problem is when we put these people on renal failure on dialysis, okay, when the dialysis machines have certain membranes in them. Okay, so I'm just going to give a brief overview of the process of dialysis just to make sure that everyone understands what this involves. Okay, so basically dialysis is something we use in people with renal failure. Okay, so the kidneys are failing, they're now not removing waste products from the blood. Now there are very dangerous waste products in the blood that are very quickly going to build up if we don't do something. One of the key ones is urea, okay, which is actually really, really toxic to cells. I mean, we've seen how it's a denaturing agent for proteins. Okay, so basically, what we need to do is somehow remove the waste products from these people's blood, and this is what dialysis is all about. So, if I draw the little person here, so this is our person who's going to be put on dialysis, and basically the idea is that we will have one tube which will take arterial blood from an artery, usually out of the arm. Okay, so let me colour this in. Here is a tube that is taking arterial blood out of an artery in the arm. And then what will happen is this will go through a special machine, the dialysis machine here, okay, and then it will come out the other side, and then we'll put it back into some vein somewhere, okay, via another tube. So this is going to go into a vein. Okay, right. Now, what then does this machine do? Well, basically this machine is just literally a tube, okay, and the tube is lined by a semi-permeable membrane. So the blood is going to go through a tube that is lined by a semi-permeable membrane. And what is this tube within? Well, it's within a much bigger vessel containing fluid. Okay, so maybe I should draw another picture of this actually. So if I draw this better, what we could have is we could have the tube actually with a thickness rather than just being represented as a single line, and it's within this machine, okay, like so. So here is the tube going through, and this membrane here is the semi-permeable membrane, and now it's surrounded by fluid, and all the time we are putting in clean fluid and taking out dirty fluid. So there is an input tube that is putting in clean fluid, okay, and there is an output tube that is taking out dirty fluid. Okay, so the idea is this. This semi-permeable membrane is the key thing here. Okay, this is going to allow small molecules such as urea to move out of the blood and into the fluid in this dialysis tank here. Okay, so this is how small waste product molecules are going to leave the blood and go into the dialysis machine, and then of course we're continuously refreshing the fluid in here, so the urea will end up going out the output tube, and then we'll put clean fluid into here. Okay, so this is how we can remove small waste products with this semi-permeable membrane. Now of course the semi-permeable membrane only has tiny little holes in it basically, so it's not going to allow red blood cells to leave the tube. They're going to stay in the tube and they're going to go back into the person. Okay, it's also not going to allow big proteins to leave. Now key to this is it does not unfortunately allow the beta-2 microglobulin to leave. Beta-2 microglobulin is simply too big to get through the semi-permeable membrane. Okay, and you might say, well, why don't you just make the holes bigger? But then you're going to lose all sorts of proteins that we don't want to take out of the blood. So it has to be a compromise, basically. This membrane does need to only be such that it allows very small molecules to leave, okay, so that we don't deplete all of the necessary proteins that are within the blood. Okay, right, so that's the principle then of dialysis, that we can remove these small waste product molecules using this semi-permeable membrane through this medium that we're continuously replacing with fresh medium. Okay, right, and of course it's working through the principle of diffusion here, that the urea will be in much higher concentrations in the blood than it will be in the surrounding fluid, and therefore it will diffuse out. Okay, right, now, why does that cause a problem then for these people with huge levels of beta-2 microglobulin? Well, basically, we used to use dialysis membranes. This semi-permeable membrane used to contain copper ions. Okay, so we used to use copper-containing dialysis membranes here. Okay, and it was something about, we think it was something about these copper 2+ containing dialysis membranes that then causes the problem with beta 2 microglobulin. So remember I've said that beta 2 microglobulin isn't going to be removed by the dialysis machine, okay, but it's not really a problem because it's not going to form amyloid fibrils on its own. But what seems to happen is when we use these copper 2+ containing dialysis membranes, some of the copper 2+ ions seem to end up going back into the blood, and they seem to combine with the beta 2 microglobulin and somehow cause the beta 2 microglobulin to misfold. So copper ions seem to be extremely good at causing the misfolding of beta 2 microglobulins, and this still isn't particularly well understood, but there is a very good correlation between using a copper 2+ containing dialysis membrane and the misfolding of beta 2 microglobulin. Okay, so then what happens is the misfolded beta 2 microglobulin that's induced by the copper 2+ ions is then going to aggregate into amyloid fibrils, and particularly these amyloid fibrils are deposited in the bone. Okay, so you get bone deposition of amyloid fibrils of beta 2 microglobulin. Okay, and what this gradually then leads to is bone erosion. Okay, which is not a good thing to have whatsoever. Okay, right, so this can lead to pathological fractures and things like that. Okay, right, so that is dialysis-related amyloidosis, and it really is related to, unfortunately, the medical treatment that we've then given people with renal failure. I mean, this medical treatment is obviously very, very much so necessary to remove the horrendous small molecule waste products, okay, but we now use membranes that don't contain copper 2+ ions. Okay, right, so that's dialysis-related amyloidosis. The final thing that I just want to say about that before we move on to Alzheimer's disease is that this is usually abbreviated down to DRA, dialysis-related amyloidosis. Okay, right, so let's now move on to Alzheimer's disease. Okay, so Alzheimer's disease then. This is the famous amyloidosis. Everyone knows that, well, not everyone knows, but it's common knowledge that Alzheimer's disease is caused by amyloid plaques within the brain. Okay, and indeed these are plaques made up of amyloid fibrils, the structure of which we've examined before. Okay, right, so let's discuss then what happens in Alzheimer's disease. So Alzheimer's disease is all about a protein called amyloid precursor protein. Okay, now this is one of these proteins that we don't actually know what the function of it is yet. Okay, so the physiological function of amyloid precursor protein isn't known. So Alzheimer's disease is all about this protein, amyloid precursor protein, usually just abbreviated down to APP. A for amyloid, P for precursor, and then the second P is for protein. Okay, now, let me show you the structure of this protein. It is a membrane protein. It's within the cell membrane of all cells, interestingly. However, Alzheimer's disease, the amyloid plaques only actually appear in the brain, and we don't really understand why that is, because the amyloid precursor protein isn't just localized in the brain. It is much, much broader. It's found all over the place, basically. Okay, but amyloid precursor protein is a transmembrane protein with a single membrane spanning alpha helix here in the middle. Now its amino terminus is extracellularly and its carboxy terminus is intracellularly. Now, part of the physiology of amyloid precursor protein seems to be that it is a protein that is cut up to make other things, okay? So it seems to be a protein that we make in order to process it into other things. Now let me explain what we think is the normal physiological way that amyloid precursor protein is processed, and then I'll show you what seems to happen in Alzheimer's disease to cause the formation of the amyloid plaques within the brain. Okay, so the normal way that amyloid precursor protein is processed involves two enzymatic cleavages, but before I just say that, I should say that there is a special portion of this protein, which I'm going to highlight in red here. Okay, so the whole thing is the amyloid precursor protein, but that red portion in the middle that I've now highlighted up, that is the portion that if we cut this up, if we cut here and produce that little red portion, this misfolds into something that can then cause amyloid fibrils, okay? So it can aggregate into amyloid fibrils, okay? And that's the portion that's eventually going to aggregate, so keep an eye on that portion. Now, in the normal, well, what we believe is the normal, healthy way to process amyloid precursor protein, what happens is you make your first cut right in the middle of this amyloidogenic portion, okay? Now, the enzymes which cut the amyloid precursor protein at this special site here, right in the middle of the amyloidogenic portion, are known as alpha secretase enzymes. Now, I want to stress that there is not just one alpha secretase enzyme, there are multiple different alpha secretase enzymes. Okay, so it's a broad class of enzymes, like earlier on when we were looking at E1 enzymes, E2 enzymes, E3 enzymes. There are loads of those enzymes, and they're defined by what they do rather than by their actual primary amino acid structure. So again, alpha secretases are a class of enzymes defined by what they do. Okay, so alpha secretase enzymes cut amyloid precursor protein in this site in the middle of the amyloidogenic portion, but the portion that isn't within the membrane spanning alpha helix. So you can see that a portion of this amyloidogenic portion extends into the membrane. So alpha secretase cuts above that in the extracellular portion. Okay, now that produces this first amino terminal fragment here. So here's the amino terminus, and then here is that beginning portion of the amyloidogenic portion. Okay, so that's one of the bits that alpha secretase produces. This is now what's known as soluble amyloid precursor protein alpha, SAPP alpha. Okay, so this is soluble, and then it's amyloid precursor protein, APP, and then alpha. Okay, and the alpha is there because it's been cut by an alpha secretase. Okay, now that doesn't seem to be harmful. We're not really sure what it does, but it doesn't seem to be harmful. Okay, now, what then happens is the remaining portion down here is then processed by another enzyme, okay, called a gamma secretase enzyme. Okay, now gamma secretase is a specific enzyme. Well, I shouldn't say it's a specific enzyme. Again, there are multiple gamma secretases, but they are massive complexes of 4 separate proteins, okay, and you can slightly vary which proteins are in that complex. So yes, there are multiple gamma secretases. So again, gamma secretase enzymes are defined by where they cut the amyloid precursor protein. Now, gamma secretase enzymes cut right at the base of this amyloidogenic portion. Okay, now there are actually 2 different sites that gamma secretase enzymes can cut amyloid precursor protein, which differ by 2 amino acids. Okay, so I'll put these here. So one of them is right at the end of the amyloidogenic portion, and one is 2 amino acids upstream of that. So gamma secretase can choose 2 different sites to cut, and the difference between them is 2 amino acids. So if it cuts at this first site, the higher one up, then this you'll get this short little red portion here. Okay, and if it cuts at the one below, that little red portion that you're going to produce will be 2 amino acids longer. Okay, so this other fragment is now being produced by the gamma secretase, which could be one of 2 different fragments depending on which site the gamma secretase chooses to cut. Okay, now, that fragment again is secreted extracellularly, and it's known as the P3 fragment. And again, its function isn't very well understood at all. Okay, so gamma secretase has these 2 different sites to cut at, and it determines, well, that they differ just by 2 amino acids. And then the final fragment that you produce is this C-terminal portion here, again, which will differ by 2 amino acids depending on where gamma secretase decides to cut. And this is then known as the AICD, okay, which stands for the amyloid precursor protein, that's the A, and then ICD always, well, nearly always stands for intracellular domain whenever you see that in the context of molecular biology. Okay, so that's amyloid precursor protein intracellular domain, and that goes into the cytoplasm of the cell. So that's the normal way that amyloid precursor protein is believed to be processed. Now, in Alzheimer's disease, what appears to happen is it doesn't get processed in this way. Instead, what happens is it gets processed in a different way, and this seems to specifically happen in the brain, okay, rather than in other tissues. Okay, so let's draw our amyloid precursor protein out again, okay, here. Okay, now, in Alzheimer's disease, the bad way of processing the amyloid precursor protein firstly involves a beta secretase enzyme rather than an alpha secretase enzyme. Now, there is just, I believe, one beta secretase that has come to dominate, okay, and we don't seem to have found another one, so I might give you the name of that specific beta secretase. So just like alpha secretase and gamma secretase, beta secretase, the name implies any enzyme which cuts at this special site. However, there does seem to only be one beta secretase, okay, and this is known as BACE1, B-A-C-E-1, which stands for beta site, so I'll write this down. So B is for beta site, and then the A is for amyloid precursor protein, and then the CE is for cleavage enzyme. So the beta site amyloid precursor protein cleavage enzyme 1. And I think that is the only beta secretase enzyme that we found as of yet. Okay, right, so when the beta secretase enzyme cuts there, what it produces is, firstly, it produces this N-terminal fragment, which is slightly shorter than the N-terminal fragment that we produced in the normal way. Okay, and this is called the soluble amyloid precursor protein, but this time not alpha, instead beta, because it's being produced by the processing through a beta secretase enzyme. Okay, so that's the soluble amyloid precursor protein beta. Then what's going to happen is the gamma secretase is then going to work on the remaining portion, and again it can cut at two different sites, okay, which differ by 2 amino acids, and then what it's going to produce is, once again, it's going to produce amyloid precursor protein intracellular domain exactly as it would have produced in the normal way of processing it, but now this red fragment that you produce now is the full amyloidogenic portion. Okay, now of course you can produce two different ones here which differ by 2 amino acids. So the shorter one, which is when the gamma secretase cuts at the top site, is then called the amyloid beta peptide 40, okay, because it's 40 amino acids long. And then the longer one is called amyloid beta 42 because it's going to be 42 amino acids long because it has those extra 2 amino acids there. Okay, now both of them are amyloidogenic, so often we just refer to amyloid beta 40/42, meaning one or the other. Okay, so they can both misfold and then form amyloid fibrils. However, amyloid beta 42 is better at it than amyloid beta 40. Okay, so this one is worse. This one is more amyloidogenic than amyloid beta 40. Okay, so these form amyloid fibrils within the brain in the extracellular fluid because, again, these amyloid beta 40 or 42 fragments are going to be secreted extracellularly. So you get extracellular amyloid plaques forming between the neurons, and these seem to be driving the neurodegenerative process of Alzheimer's disease. Okay, so they somehow cause neurons to start degenerating, and this leads to the dementia that is seen in Alzheimer's disease. Okay, so it causes dementia. Okay, and what does dementia involve? Well, the three main things that happen in dementia are cognition goes down, your ability to think and problem solve and things like that. Your personality starts to change, okay, and also memory is lost. Okay, so those are the three main characteristics of dementia, memory lost. Right. Okay, so last thing I would just like to say is that most cases of Alzheimer's disease are sporadic. Okay, we cannot find any germline mutations that would suggest that you should have Alzheimer's disease. Okay, however, there are forms of Alzheimer's disease which are familial, okay, where we can find mutations in your germline genome which predispose you to having Alzheimer's disease. And by the way, Alzheimer's disease is often just abbreviated down to AD for short. Okay. Right, so some people do have familial Alzheimer's disease where we can look at their genome from birth and say that they are at risk of developing Alzheimer's disease, and generally they end up developing Alzheimer's disease earlier than a sporadic case of Alzheimer's disease would develop. Okay, now generally what happens is you either have mutations in the amyloid precursor protein, okay, which predispose it to being processed in this bad way rather than in the good way, or alternatively predispose it to being cut by the gamma secretase in this second site rather than the first site. So if you have mutations that cause the amyloid precursor protein to be more likely cut at this second site by the gamma secretase, then that means you're going to generate more amyloid beta 42 than 40 when amyloid precursor protein is processed in this bad way, okay? And this one's more amyloidogenic. You'll develop Alzheimer's disease more rapidly if you're producing lots of that rather than lots of this, okay? So that's one of the things that could underlie early onset Alzheimer's disease in this familial way. In addition, you can have mutations in proteins known as presenilins. Okay, now the presenilins are parts of the gamma secretase enzymes. Okay, so remember I told you that gamma secretase isn't just one protein. The gamma secretase enzymes are complexes of 4 proteins. One of the key proteins that's within the gamma secretase complex are proteins known as presenilins. Okay, and there are multiple different types of presenilins, which is how you end up with multiple different gamma secretases, because they can have different presenilins and then therefore end up different gamma secretases. Okay, but if you have mutations in the presenilins of the gamma secretase enzymes, this can make the gamma secretase again more likely to cut the amyloid precursor protein at the second site and therefore produce more amyloid beta 42 when amyloid precursor protein is processed in this bad way here that leads to the formation of amyloid beta plaques. Okay, right, so that's a little bit of an introduction to the pathology of Alzheimer's disease. Again, it's a protein folding disorder. These amyloid beta fragments, the amyloid beta 40 and 42, they misfold and then they form these protein aggregates. And that's then what drives the neurodegenerative process of Alzheimer's disease. Okay, right. So now we're going to look at a disease that isn't generally thought of as an amyloidosis disease, but it's starting to gradually be realized how potentially amyloidosis plays a really important part in it. Okay, and this is the pathology of type 2 diabetes mellitus. Okay, a chronic metabolic disease. So T2DM stands for type 2 diabetes mellitus. So I'll just briefly outline the pathology of diabetes mellitus, and then we can see where the formation of amyloid fibrils due to protein misfolding is going to play a role here. Okay, and I'll just give the answer away now for those who know something about type 2 diabetes mellitus. It's believed to play a role in beta cell dysfunction that occurs as part of the pathology of type 2 diabetes mellitus. Okay, so what I'm just going to briefly do is outline normal glucose homeostasis, and then what we can see is what happens in type 2 diabetes mellitus. So firstly, I probably should just say it's a metabolic disorder that affects your ability to handle glucose correctly. Okay, right, so let's have a look at normal glucose homeostasis. So within the pancreas is the first place to start. So if we draw a pancreas here, okay, most of the pancreas is exocrine tissue and is involved in producing the digestive enzymes which are secreted into the intestine, okay, those enzymes which get blocked in cystic fibrosis. However, there are little patches of the pancreas which are endocrine in function, i.e. they're secreting things into the blood rather than outside of the body as the digestive enzymes are secreted. Okay, and these little clusters are known as islets of Langerhans. Okay, and these are extremely important in the normal homeostasis of blood glucose concentration. So let's draw one of these now bigger here. So this is an islet of Langerhans. Now, there are many different types of cells within the islets of Langerhans, but the two that are going to be really important for us are alpha cells and beta cells. So let's say this one here is an alpha cell. Okay, and this one here is a beta cell. Okay, now let's colour them in different colours. So we'll have the beta cell in pink here, okay, and then we'll have the alpha cell in green. Now, they secrete two utterly opposite hormones, okay, so the alpha cells secrete glucagon, okay, and the beta cells secrete insulin, and they both respond to blood glucose concentration, okay, so if blood glucose concentration is very high because you've just eaten a meal, beta cells secrete loads of insulin and alpha cells secrete hardly any glucagon. At normal blood glucose concentration, okay, the fasting blood glucose concentration, which incidentally is around 5 millimolar in the millimolar units, or if you're American, that's 90 milligrams per deciliter. The way of converting between those two units is merely multiply by 18. Okay, so if you've got normal fasting blood glucose concentration, then the beta cells will be secreting hardly any insulin, and the alpha cells will be secreting loads of glucagon. Okay, so whenever talking about normal blood glucose homeostasis, then we need to split it into two separate scenarios. We need to split it into the fasting blood glucose homeostasis, where you've not just eaten, okay, so when you haven't eaten for a while, and how blood glucose concentration is being maintained there. And then we also need to split it into postprandial glucose homeostasis, which is glucose homeostasis just after you've consumed a meal. Okay, so let's start off with fasting blood glucose homeostasis. So how do we maintain a normal blood glucose concentration when we are fasting? So we can rule the intestine out, which makes it far more simple. Okay, so the intestine is not giving us more glucose. Okay, so what is happening? Well, if we draw the blood here, peripheral tissues are taking glucose out of the blood and using it for respiration. Okay, and the key peripheral tissue that we'll use as an example is the brain. Okay, so here's the brain taking glucose out of the blood and using it for respiration. Okay, so if we're going to maintain a constant blood glucose concentration, then we need to be putting in to the blood the same amount of glucose as the peripheral tissues are taking out. Okay, and this is performed by the liver. Okay, so I'll put the liver here. Okay, so at normal fasting state, the liver is putting in the same amount of glucose into the blood as the peripheral tissues are taking out, and that's how we maintain a normal blood glucose concentration. Now, the liver, the way the liver is coordinated is by insulin and glucagon concentration. So the liver basically is a little slave here. Okay, it isn't measuring blood glucose concentration and deciding how much glucose it should be putting in. No, it listens to the insulin and the glucagon. So the liver is very stupid and has to just obey the orders of the alpha cells and the beta cells, which tell it how much glucose to be putting into the blood. So, in a normal healthy person where blood glucose concentration is perfectly, blood glucose handling is perfectly healthy, okay, at this normal fasting blood glucose concentration, what will happen is the alpha cells will be secreting lots of glucagon, so glucagon will be very high, okay, and the beta cells will not be secreting that much insulin, so insulin will be very low. Okay, but not zero. That's really important to understand, not zero. Okay, so you still have some insulin. Okay, and both of these will be acting on the liver. Now, glucagon is telling all of the hepatocytes to throw out loads and loads of glucose into the blood. So this says, yes, keep going, put more and more glucose in. Okay, whilst the insulin is basically a cautionary signal. It says, you know, this is a little bit over the top, tame it down a bit. Okay, so the glucagon says chuck in loads, the insulin says no, chuck in a bit, do a bit of what the glucagon says but tame it down. So basically the insulin signal is doing the opposite to the glucagon signal and tames the glucagon signal down a bit basically. Okay, and this is important because if you lose the insulin signal, okay, now the glucagon signal is totally over the top. The liver responds over the top because it's no longer being counterbalanced by the insulin signal, and you end up chucking in far too much glucose into the blood, more than the peripheral tissues use, and therefore you tip this balance in favour of blood glucose rising. Okay, so Then in a normal healthy person, the insulin signal, however, is counterbalancing the glucagon signal to a certain extent, and that keeps it beautifully in balance with you secreting the right amount of glucose into the blood to meet the needs of the peripheral tissues. Okay, so that's normal fasting blood glucose homeostasis. Now let's talk about what happens in postprandial glucose homeostasis, so following a meal. Okay, so the key thing that's happening now is that the intestine, which I'll do a little picture of here. So here's the stomach, and then here is a portion of the duodenum, and I'll stop there, I won't draw the entire intestine out. Okay, so the intestine is now chucking glucose into the blood. Okay, and it's chucking a huge amount of glucose into the blood. So you've eaten a very carbohydrate-rich meal, you've eaten loads of bread or potato or something along those lines. You've got loads of glucose coming into the blood. Okay, what happens now? Well, basically, the beta cells now secrete loads of insulin. Okay, so insulin goes up hugely, and glucagon secreted by the alpha cells goes right down. Okay, so glucagon goes right down. Now what happens is this acts on the liver. Okay, so if I draw the liver here, now the insulin signal is stronger than the glucagon signal, and what the insulin signal now does is it tells the liver to take glucose out of the blood and store it, basically, and then the liver can release this glucose later on in the fasting state. Okay, so the liver starts doing the exact opposite, taking glucose out of the blood and storing it. That's not just the liver that the insulin acts on. Okay, so both of these signals are causing the liver to now start taking glucose out. The fact that the glucagon signal's gone down and the insulin signal's gone up now causes the liver to reverse its activity. But insulin actually has effects on other tissues as well, specifically, really importantly in fact, the skeletal muscle cells. Okay, now this is really important. This is actually more important than the effect on the liver as far as coping with postprandial glucose rise is concerned. Okay, so let's say this is a piece of skeletal muscle here, and I need to add some colour onto this picture to make it look more interesting. Okay, so here is the skeletal muscle, and you know, you have a huge amount of skeletal muscle in your body. A huge amount of your body mass is made up of skeletal muscle. Okay, so what insulin actually does is when it goes up, it works on skeletal muscle cells and causes them to start taking glucose out of the blood and storing it as glucagon, which can then be used when the skeletal muscle contracts later on. And do not underestimate how important this is. Okay, this is the main way that you take glucose out of the blood when you've just eaten a meal. It goes into the skeletal muscle. The amount that's moving into the skeletal muscle dwarfs the amount that's moving into the liver. Okay, finally, insulin also works on adipocytes, okay, fat cells. So here is our fat cell with a massive great store of fat in the middle there. Okay, so this is an adipocyte. And insulin being high causes the adipocytes to take glucose out of the blood and then convert it into fat molecules. Okay, so that's why when people say, oh, you have to eat fatty foods to become fat, it's not true. Okay, carbohydrate-rich foods can be converted to fat by the body easily. Okay, so glucose can be converted into fat. Sugar can be converted into fat. Okay, so that's the way then that insulin deals with the glucose concentration going through the roof because you've just eaten a meal. Okay, so normal blood glucose homeostasis can be summed up with a nice little graph then. Okay, so on the y-axis we will have our blood glucose concentration, and on the x-axis we'll have time here. Okay, so in the normal fasting state then, blood glucose remains at this beautiful level of 5 millimolar, which is the equivalent of 90 milligrams per deciliter. Okay, and then what happens is when you eat a meal, you get a spike in blood glucose like this, which then comes nicely back down because of the beautiful postprandial glucose homeostasis mechanisms, and then you return back to the fasting blood glucose homeostasis. Okay, right, so that's normal blood glucose homeostasis. And by the way, this sort of a spike has a special name. This is what's known as a postprandial, okay, which just means after eating glucose spike, or just a postprandial spike. Okay, so postprandial glucose spike. Right, okay, so now let's discuss type 2 diabetes mellitus. So the key features of type 2 diabetes mellitus then are that you have fasting hyperglycemia. Okay, now what that means is that your blood glucose concentration is too high even in the fasting state. Okay, that's usually defined as being greater than 7 millimolar. Okay, in the fasting state. Okay, so just multiply that by 18 to find out what that is in milligrams per deciliter. In fact, we'll do it. 70 is 7 times 10. Okay, 7 times 8 is 56. Okay, so adding those two together, you get 126 milligrams per deciliter. So in the other units, it's greater than 126 milligrams per deciliter. Okay, right. So drawing that on the graph, it'll look like this. You have raised blood glucose in the fasting state. Okay, so that's one of the features of type 2 diabetes mellitus. And then the other one is that you have horrendous postprandial glucose spikes. So your postprandial glucose spikes are going to be taller, firstly. Your blood glucose is going to go more out of control, and they're also going to be much, much broader. Okay, so what it's going to look like when you have a meal is something like this, basically. Dun, 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 dun. Okay, I'm ruining the picture like that. Okay, so it's going to be massive, great postprandial glucose spikes. Your postprandial glucose control is terrible. Okay, like so. So that's what type 2 diabetes mellitus involves, both the fasting hyperglycemia and the taller, broader postprandial glucose spikes. I'll just put spikes there. Okay, right, and this is not good at all. That hyperglycemia in this way causes all sorts of pathological features, which we're not going to go through here, but it causes microvascular disease and macrovascular disease, and it can result in horrible, horrible pathologies. Okay, so uncontrolled diabetes is not good. So now let's have a look at how this actually occurs. What causes type 2 diabetes mellitus? Well, originally what happens is you get peripheral insulin resistance. So insulin resistance is believed to be the starting point for this. Now, what causes insulin resistance? We don't know, but the main risk factor for insulin resistance is obesity, okay? And that's why people often refer to the diabesity epidemic, okay, which is the mixture of having diabetes, type 2 diabetes, and being obese, because they almost invariably go together. Obesity seems to cause type 2 diabetes mellitus because it drives peripheral insulin resistance. Okay. Also, lack of exercise, which often goes with obesity as well. So lack of physical activity seems to be a major risk factor for developing insulin resistance. Okay, so what is meant by insulin resistance? Well, basically, it just means that your cells become less good at responding to insulin. Okay, so remember which cells respond to insulin. Okay, it is the liver cells. We'll start with those. The liver cells, remember, when insulin is high, they are responding to this insulin, and they take glucose out of the blood and store it as glucagon. In addition, the skeletal muscle cells respond to high insulin and take blood glucose out of the blood and store it as glucagon. And the same for the adipocytes, they take blood glucose out of the blood and store it as fat molecules. Okay, so if these tissues all stop responding to the insulin, or at least tone down how much they respond to the insulin, then that makes sense that we will get much higher postprandial glucose spikes, and also much broader. It'll take us much longer to return blood glucose back to normal because these tissues just aren't responding to the glucose properly. Okay, in addition, let's think about the fasting blood glucose concentration. Again, I told you that the insulin signal is important for taming the glucagon signal. If the insulin signal is now reduced, not because insulin's gone down, but because its effect on the liver cells has been reduced, okay, the liver is now insulin resistant, okay, then the insulin signal, the effect of insulin on the liver cells is going to be reduced, and it's no longer going to counterbalance the glucagon signal in the way that it needs to. So now the liver is going to respond to the glucagon signal in full, and it's going to chuck out far too much glucose into the blood, more than the peripheral tissues can use, and that's why blood glucose is elevated in the fasting state. So that's why you get fasting hyperglycemia. Okay, now, initially, however, insulin resistance doesn't cause a problem because you get a phenomenon known as beta cell compensation. Okay, so basically what this involves is, okay, insulin is now less efficacious at the peripheral tissues, okay, the liver, the skeletal muscle, and the adipocytes. So what are the beta cells going to do? They start chucking out more insulin, okay? If the cells, if the peripheral tissues are not responding to the insulin as well, all we need to do is make more of it, and then we can get the insulin signal back up to what it should be, okay? So initially, when you start developing the insulin resistance, it doesn't cause a problem because the beta cells compensate and they just produce more insulin. So initially you don't get hyperglycemia, you don't get broader postprandial glucose spikes or taller postprandial glucose spikes. Instead what you get is hyperinsulinemia. Okay, so your insulin levels within the blood go up. Okay, so hyperinsulinemia both in the fasting state and in the postprandial state. Okay, however, then unfortunately what happens is the beta cells stop doing this, okay? You get what's known as beta cell dysfunction. Now originally, when this was first discovered, people originally called it beta cell exhaustion because the idea was that beta cells have been working so hard and they just get exhausted of doing it, okay? And then they suddenly stop producing as much insulin. Okay, now we don't think it's due to exhaustion of the beta cells. We don't think they're getting tired, so we call it beta cell dysfunction instead. So basically what starts happening is beta cells in the islets of Langerhans stop secreting insulin. Okay, some of them even start dying. Okay, so you get a lot of beta cell dysfunction in the pancreas. Okay, and this then causes insulin secretion to drop. So the hyperinsulinemia goes away, and therefore the compensation for the peripheral insulin resistance is lost, and then the insulin resistance starts having an effect. So it causes the fasting hyperglycemia that we've just discussed and the too tall and too broad postprandial glucose spikes. Okay, right, and it's this that we are going to study because this is believed to be caused by amyloidosis. Okay, the formation of amyloid fibrils near the beta cells, basically. Okay, so what I should probably just say is that this is still controversial, okay? What causes beta cell dysfunction in type 2 diabetes mellitus is still controversial. However, there is a leading theory, and the leading theory is that it might be due to amyloid fibrils forming near the beta cells. We know that amyloid fibrils do form near the beta cells. What we don't know is they are what kill the beta cells, okay? But it's one of the leading theories. So let's have a look at this. So this involves a protein known as IAP, okay, IAPP. Okay, now what does this stand for? This stands for islet, that's the I, okay, and then amyloid is the A, and then PP is for polypeptide, so islet amyloid polypeptide. Okay, now islet amyloid polypeptide also has another name. You can also hear it referred to as amylin. Okay, now, this is a protein whose function we do not know, but which is secreted by the beta cells of the islets of Langerhans. So remember, the beta cells are the ones which secrete insulin, okay, and in fact, the islet amyloid polypeptide is secreted by the beta cells whenever they secrete insulin. So whenever they secrete insulin, they also secrete islet amyloid polypeptide, so IAPP is also secreted. Okay, now IAPP is not a brilliant protein. It has a tendency to misfold and aggregate into amyloid fibrils. Now, in the pathology of type 2 diabetes mellitus, the peripheral insulin resistance causes beta cell compensation initially. The beta cells start secreting far too much insulin to compensate for the peripheral insulin resistance. That means that they also secrete far too much IAPP, islet amyloid polypeptide. Okay, the idea then is that the islet amyloid polypeptide builds up in the extracellular fluid surrounding the beta cells, and then in the extracellular fluid it starts misfolding, forming amyloid fibrils and then forming amyloid plaques of islet amyloid polypeptide near the beta cells. So here is one of these amyloid plaques that's been formed by the islet amyloid polypeptide. The idea then is that the islet amyloid polypeptide amyloid plaques kill the beta cells, and this then leads to the beta cell dysfunction. So the over-secretion of insulin leads to the over-secretion of the islet amyloid polypeptide, and that then leads to the formation of these amyloid plaques containing islet amyloid polypeptide in the vicinity of the beta cells, and that then leads to the death of the beta cells, which leads to beta cell dysfunction, which means that you can no longer have beta cell compensation for the peripheral insulin resistance, and then the peripheral insulin resistance causes the type 2 diabetes mellitus. So that by the time you actually get type 2 diabetes mellitus symptoms, i.e. the fasting hyperglycemia and the too large postprandial glucose spikes and all of the actual pathological symptoms that are associated with that, okay, you've actually already gone through the peripheral resistance, already gone through the beta cell compensation, and already gone through the beta cell dysfunction. So those of your beta cells are already dead, basically. Okay, right, so that's the potential involvement of gain-of-function protein misfolding diseases in the pathology of type 2 diabetes mellitus. In the next video, what we'll do is we'll turn our attention onto prion diseases, and then finally onto serpinopathies.